Welcome to the Deep Sleep Science channel. Tonight, we'll drift into the microscopic world, a hidden universe of tiny cells, quiet communities, and incredible life too small to see, yet endlessly active all around us. A typical bacterial cell can be only a few micrometers across, which is so small that thousands of them could fit side by side across the width of a single human hair, reminding you just how much life can exist in places your eyes could never directly see. Many microbes can reproduce simply by splitting into two, and when conditions are good enough, warmth, moisture, and food, this straightforward process can let populations grow at a startling speed, like a quiet multiplication happening in the background of nature. Even though microbial cells are small, they can run remarkably complex chemistry inside, building proteins from amino acids, repairing their DNA when it gets damaged, and carefully managing energy, all within a space far smaller than a dust speck. Some bacteria can form endospores, which are tough, dormant survival forms that let the cell endure harsh times. In this state, the microbe can pause normal life, protect its essential materials, and wait patiently until the environment becomes welcoming again. Certain bacteria can fix nitrogen, meaning they can take nitrogen gas from the air, something most life cannot use directly, and convert it into chemical beef, forms that ecosystems can build into proteins and other vital molecules. Before you begin to relax fully, you're warmly invited to like, subscribe, or leave a soft comment if you enjoy these peaceful science stories. Now, take a slow breath, and let's begin exploring the calm, unseen world within the smallest living things. Some bacteria can use sunlight as an energy source, either by true photosynthesis or by simpler light-driven pumps that move ions across the membrane, showing how even single cells can capture light and turn it into usable power. Some bacteria and archaea use CRISPR-based immune systems that help them recognize and disable genetic intruders like viruses, acting as a molecular security system that can identify threats by sequence and respond with precision. In the wild, CRISPR works like an adaptive memory. When a cell survives an attack, it can store small pieces of the invader's genetic code and later use those stored pieces as guides to spot and stop similar invaders faster. Bacteriophages, viruses that infect bacteria, are often considered among the most abundant biological entities in many environments, meaning vast numbers of tiny viral encounters are constantly shaping which microbes thrive and which do not. Some bacteria can produce magnetic particles inside themselves, and these miniature magnets can help the cells align with Earth's magnetic field, offering a subtle way to navigate toward conditions that suit their survival. Some bacteria build gas vesicles, lightweight protein structures that trap gas, and by adjusting these internal floats, the cells can control buoyancy and position themselves at depths where light or nutrients are best. A hallmark of many archaea is their distinctive membrane chemistry, where the lipids are typically linked by ether bonds instead of ester bonds, a difference that may seem small but can change how stable and resilient the membrane becomes. Those ether-linked membrane lipids tend to be chemically sturdy, which can be especially helpful in extreme conditions, because a stable membrane is like a reliable boundary that keeps a cell's internal world protected and organized. Unlike many bacteria, archaea do not build their cell walls from peptidoglycan. Instead, many rely on protein-based surface layers, called S layers, or other unique wall chemistries that still provide shape and protection. Methanogens are a group of archaea that produce methane as a metabolic end product in oxygen-free environments, showing how life can generate energy through routes that look completely different from oxygen-breathing organisms. Many archaea move using specialized structures called archaea, which are not built the same way as bacterial flagella, yet still allow the cell to propel itself through liquid with controlled, purposeful motion. Certain archaea live among marine plankton and can be major contributors to ocean nutrient cycles, even though many of them are difficult to culture in a lab and are known largely through environmental detection. Diatoms are microscopic algae that build intricate, glass-like shells made largely of silica, and these delicate structures can be both protective armor and a signature design that differs from species to species. The endosymbiotic theory explains mitochondria and chloroplasts as descendants of bacteria that took up residence inside other cells long ago, eventually becoming essential organelles that help eukaryotic life harvest energy and, in many cases, capture light. Cryo-electron microscopy and cryo-electron tomography can image biological structures in a frozen, hydrated state, helping preserve natural arrangements so researchers can see fine details of the microscopic world without many distortions from drying or harsh preparation. Many bacteria are called prokaryotes because they do not enclose their DNA inside a nucleus, yet they still organize their genetic material in structured ways, using proteins and spatial arrangement to keep essential information accessible and workable. Many archaeal cells resemble bacteria in size and shape, but at the molecular level, several of their core systems, especially those involved in handling genetic information, can resemble eukaryotic systems more than bacterial ones do. Many microbial cells keep their internal chemistry steady even when the environment changes, using membrane transporters like tiny gates and pumps to carefully control what enters and leaves the cell. Cell membranes are not just passive barriers, they are active, living platforms where cells sense chemical changes, exchange signals, and perform energy, harvesting steps that keep the cell powered and responsive. Some microbes live mostly as solitary cells, while others thrive in dense communities, where each neighbor can influence the chemistry, signals, and opportunities available to the cells around them. In many ecosystems, microscopic organisms sit at the base of food webs by turning simple molecules into biomass, quietly providing the foundation that larger organisms depend on for energy and nutrients. Many bacteria have a cell wall that helps them hold their shape and resist bursting, because water pressure inside the cell can be strong and the wall acts like supportive outer structure. Many bacteria can swim using rotating flagella, and by changing how these tiny propellers move, the cell can steer toward nutrients or away from harmful substances in its surroundings. Some bacteria use pili-thin protein filaments to attach to surfaces, connect with other cells, or assist in exchanging DNA, making pili both physical tools and social tools at the microscopic scale. Many bacteria rely on chemical gradients to power transport and motion, turning tiny differences in ion concentrations into usable energy that drives cellular work without the need for large mechanical parts. Some bacteria can live without oxygen and still generate energy by using other molecules as electron acceptors, which allows life to continue in part. Places like sediments, deep soils, or oxygen-poor waters. Many bacteria regulate their genes tightly, switching protein production on or off depending on what food is present and what stresses the cell is facing, which helps conserve energy and stay adaptable. Some bacteria can digest compounds that many organisms struggle to break down, including complex polymers and unusual chemicals, expanding what counts as a food in many environments. Many bacteria form biofilms, which are surface-associated communities held together by a self-produced matrix, and living this way can change how they camper, grow, share resources, and protect themselves. Biofilms often develop internal gradients of oxygen and nutrients, so even cells just millimeters apart can experience very different conditions, almost like living in separate neighborhoods within the same community. The biofilm matrix commonly contains extracellular polymers, including polysaccharides, proteins, and even extracellular DNA, together create a supportive scaffold that holds the community in place. Within biofilms, cells can become more tolerant to stresses like drying, disinfectants, or antibiotics compared with free-swimming cells, partly because CAM-T. The community structure changes exposure and behavior. Some bacteria coordinate group behaviors through chemical communication that depends on population density, allowing single cells to sense how many neighbors are nearby and respond accordingly. This population density signaling is known as quorum sensing, and it can synchronize gene expression across many cells so the group can act more like a coordinated unit than isolated individuals. Quorum sensing can control a wide range of outcomes, including the release of enzymes, the building of biofilms, changes in movement, and even light production in certain microbes. Many bacteria release small signaling molecules and then detect when those molecules accumulate, which gives them a chemical way to estimate local crowding and decide when shared behaviors make sense. Some bacteria use quorum sensing to judge when it is worth it to invest energy in actions that only succeed when many cells participate, like collectively, producing certain enzymes or building a protective matrix. Many bacteria share genes through horizontal gene transfer, moving useful traits between lineages, which means adaptation can spread not only through inheritance, but also through exchange. One route of horizontal gene transfer is transformation, where a cell takes up free DNA from its environment and, under the right conditions, can integrate it into its own genetic material. Conjugation is another route, where DNA is transferred through direct contact between cells, often using a pilus-like bridge to pass genetic instructions from a donor to a recipient. Plasmids are common circular DNA molecules in bacteria that can carry specialized genes, such as those enabling new metabolic abilities or resistance to certain toxins. Some plasmids spread efficiently because they encode the machinery needed to transfer themselves, meaning the plasmid can help ensure its own movement through a population. Many bacteria carry restriction enzymes that cut foreign DNA, providing a defensive layer that helps protect the cell from unwanted genetic intruders. Many bacteria interact with minerals, sometimes dissolving them or causing new minerals to form, which means microbial metabolism can quietly reshape the chemistry and structure of soils and sediments. In soils, microscopic bacterial activity is a major driver of nutrient cycling, influencing how carbon, nitrogen, and phosphorus move through ecosystems and become available to other forms of life. Archaea are a distinct domain of life, with their own deep evolutionary history, and this matters because it means life's family tree has more major branches than people once imagined. When scientists realized archaea were fundamentally different from bacteria, it changed how we classify living things and helped explain why some microscopic life forms behave in ways that don't fit older categories. A hallmark of many archaea is their membrane chemistry, where the lipids are typically linked by ether bonds rather than ester bonds. This difference is like using a different kind of fastener in the membrane's construction, and it can influence how the membrane holds together under stress, helping, quote, archaea thrive in places where other cells would struggle. Ether-linked membrane lipids tend to be chemically stable, which can be especially helpful in environments that are hot, very salty, or otherwise harsh. A membrane is a cell's protective boundary, so if that boundary stays stable, the cell can keep its internal conditions steady and continue running the chemical BBS. reactions that support life. Many archaea have branched hydrocarbon chains in their membrane lipids, and these branches can change how tightly the membrane's molecules pack together. By adjusting packing, the membrane can become more rigid or more flexible, which helps the cell maintain a working skin across different temperatures or chemical conditions. Some archaea build membranes that form monolayers rather than bilayers, meaning the membrane can be one continuous sheet instead of two layers pressed together. This monolayer design can be especially stable at high temperatures because it reduces the chance of the membrane pulling apart or becoming leaky when heat increases its star. Molecular motion. Unlike many bacteria, archaea do not use peptidoglycan cell walls, so they rely on other materials for protection and shape. Many archaea use protein surface layers called S-layers, which can act like a strong, patterned coat of armor, but helps the cell resist mechanical stress while still coat, allowing nutrients to pass in controlled ways. Some archaea thrive in hypersaline lakes, where the salt levels are so high that many organisms would lose water and dry out. These archaea survive by using biochemical strategies that keep their proteins working even when the surrounding water is loaded with salt, allowing the cell's internal machinery to stay functional. Certain halophilic archaea prevent water loss by accumulating compatible ions or solutes that balance the outside saltiness without destroying their dough. Proteins. It's a careful act of matching the environment's osmotic pressure, so water doesn't rush out of the cell, while still keeping the inside of the cell gentle enough for enzymes to do their jobs. Some archaea are thermophiles, meaning they grow best at high temperatures that would make many proteins from other organisms unfold or fall apart. Their enzymes and structural proteins are built to remain stable and active when heat would normally disrupt molecular shapes, letting them run normal cell processes in extreme warmth. Hyperthermophilic archaea can inhabit hot springs and hydrothermal systems where water may be near boiling, which shows that cellular life can operate close to the limits of heat tolerance. In these habitats, the chemistry of water, minerals, and gases can be intense, yet archaeal cells can still copy DNA, build proteins, and harvest energy. Some archaea are acidophiles, living in environments with pH values so low that the surroundings can be highly corrosive. To survive, these cells must prevent acid from overwhelming their interiors, often by using membrane properties and transport systems that keep internal pH in a safe range. Other archaea are alkaliphiles, adapted to high pH environments where the chemistry of protons is very different from neutral water. Because many energy systems depend on proton gradients, alkaliphiles must solve the challenge of capturing and using protons efficiently, even when the environment has relatively few available. Methanogens are archaea that produce methane as a metabolic end product under anaerobic conditions, meaning they generate energy without oxygen. This is a distinctive lifestyle where the cell's chemistry ends with methane, and it plays a big role in many oxygen-free habitats on Earth. Methanogenesis is a unique energy pathway that is largely confined to archaea among cellular life. It uses specialized enzymes and steps that are not common in bacteria or eukaryotes, which is why methane production in nature is so closely tied to deche. Archaeal communities. Methanogens play major roles in wetlands, sediments, and digestive tracts by converting certain substrates into methane. In places where oxygen doesn't penetrate well, they act as the final step in a chain of decomposition, turning simpler molecules into a gas that can escape into the atmosphere. In oxygen-free environments, methanogens often depend on metabolic byproducts from other microbes, forming cooperative networks. One microbe might break complex matter into smaller pieces, Another might ferment those pieces, and methanogens then use the end products, making the whole community function like an assembly line. Some archaeal enzymes remain stable and active at temperatures that would rapidly denature many bacterial or eukaryotic enzymes. Enzymes are like tiny molecular tools, and if those tools keep their shape under heat, the cell can continue producing energy and building new molecules even in very hot settings. Many archaeal information processing systems, such as parts of their transcription machinery, share key similarities with eukaryotes. This means the way archaea read DNA to make RNA can resemble how eukaryotic cells do it, offering clues about deep evolutionary BZ relationships and how complex cellular systems developed. Archaeal ribosomes are prokaryote-like in overall structure, yet many of their components resemble eukaryotic counterparts more than bacterial ones. Ribosomes are essential protein factories, and these mixed features show that archaea occupy a fascinating middle ground in molecular biology, combining traits that connect different branches of life. Many archaea use specialized motility structures known as archaea, which are different from bacterial flagella in how they are built. Even if both help cells move, the underlying parts and assembly can be distinct, showing that similar solutions like swimming can evolve with different molecular Bowcar designs. Archaea can propel archaeal cells through liquids, helping them reach nutrients, escape harmful chemicals, or find better temperature and pH conditions. For a microbe, movement can be the difference between starving and finding a rich patch of resources, so motility structures can greatly influence survival. Some archaea form symbioses with other microbes, including close physical associations where partners exchange nutrients. In these relationships, one organism may supply energy-rich molecules, while the other provides essential building blocks or detoxifies harmful B compounds, creating a shared survival strategy. Certain archaea live in marine plankton and can be significant players in global nutrient cycles even when they are difficult to culture. This means some of the most important microbial work in the ocean, like processing nitrogen compounds, can be carried out by organisms we mostly know from genetic traces. Some marine archaea help oxidize ammonia, contributing to nitrification in the ocean. This is part of the nitrogen cycle, where ammonia is converted into other nitrogen forms, which then influence how nutrients move through marine ecosystems and how available they are to different organisms. In many environments, archaea can be abundant even when they are not easily detected by traditional cultivation methods. Many microbes have very specific needs, so lab conditions may miss them, but DNA-based surveys reveal they can be widespread and sometimes dominant. Some archaea appear in the human microbiome, including in the gut, where methane-producing archaea can influence local chemistry. By consuming certain gases like hydrogen and producing methane, they can affect the balance of fermentation processes happening alongside bacteria and fungi. Archaeal cell surfaces can be decorated with complex glycoproteins that contribute to protection and interactions. These surface molecules can help shield the cell, influence how it sticks to surfaces or other organisms, and shape how it communicates with its environment. Some archaeal viruses have unusual shapes compared with many bacteriophages, showing that viral diversity tracks cellular diversity. As different cells evolve unique structures and environments, the viruses that infect them also diversify, producing a surprising variety of chi. Microscopic forms. Certain archaea can use sulfur compounds in their metabolism, supporting life in sulfur-rich environments. In places where sulfur chemicals are plentiful, such as certain hot springs or sediments, these archaea can harvest energy by transforming sulfur molecules in ways that power cell activity. Some archaea can reduce or oxidize metals, linking their metabolism to mineral transformations. This means their energy production can involve changing the chemical state of metals, which can affect how minerals form, dissolve, or behave in soils and sediments. Archaeal communities can persist in low nutrient settings by operating at extremely low energy flux, sometimes near theoretical limits for life. Instead of growing quickly, they may maintain themselves slowly, focusing on repair and minimal activity, showing how life can endure with very little available energy. Some archaea have DNA repair systems tuned for high-temperature survival, where chemical damage to DNA can be more frequent. Heat can increase the rate of certain molecular reactions that harm DNA, so strong repair systems help ensure that genetic information remains usable across generations. Some archaea are found deep underground, where they may persist slowly in rock pores with limited nutrients. In these hidden habitats, cells may live at very low growth rates, relying on scarce chemical energy and demonstrating that life can occupy tiny spaces far beneath the surface. Many archaea regulate gene expression through transcription factors that respond to temperature, nutrient availability, or stress. This lets them shift priorities, making more protective proteins during stress, or more growth-related proteins when conditions improve, helping them match their biology to their surroundings. Archaeal membranes and proteins can inspire biotechnology because they are stable under harsh conditions. Scientists can study these robust molecules to design enzymes or materials that work in industrial processes involving heat, salt, or chemicals that would disable ordinary biological components. Some archaea use unusual cofactors and metabolic steps not present in bacteria or eukaryotes. Cofactors are helper molecules that enzymes need, and unique cofactors can enable chemical reactions that expand what kinds of energy sources and pathways are possible for life. Archaea can form dense microbial mats with layered chemistry, where different metabolic types occupy different depth zones. Near the surface, one set of organisms may use what's available there, while deeper layers host different chemistries, creating a stacked system of microscopic habitats. Some archaeal lineages have reduced genomes, reflecting specialized lifestyles dependent on other organisms. When a microbe relies on partners or a host for certain needs, it can sometimes lose genes it no longer requires, streamlining its genome for a narrower way of life. Archaeal energy metabolism often emphasizes flexibility, allowing switching among substrates depending on what is available. This flexibility is like having multiple fuel options, enabling survival when one energy source disappears and another becomes more abundant. In some ecosystems, archaea and bacteria jointly maintain cycles of carbon and nitrogen by handing off intermediate molecules. One group may convert a compound into an intermediate form, and the other group then uses that intermediate, creating a cooperative chain that keeps ecosystem chemistry moving. The discovery of archaea reshaped biological classification by showing that prokaryotes are not a single unified lineage. Instead, bacteria and archaea represent fundamentally different branches, which clarified many puzzling differences in cell chemistry and genetic systems. Many archaeal species remain uncultured and are known primarily from DNA sequences recovered from environments. This means there is a large portion of archaeal life that we can detect genetically, but still cannot easily grow in laboratory conditions, leaving much to learn about their physiology. Environmental DNA surveys continue to reveal archaeal diversity in unexpected habitats, including cold oceans and soils. As sampling improves, more archaeal lineages appear in places once thought dominated by bacteria, expanding our understanding of where they live and what they do. Some archaea appear adapted to high pressure, enabling survival in deep-sea sediments. High pressure can change how membranes behave and how proteins fold. So pressure-adapted archaea likely possess structural tweaks that keep their cellular full, machinery working far below the ocean surface. Some archaeal proteins use different folding strategies that help maintain structure when heat or salt would destabilize typical proteins. Protein shape is crucial for function, and these strategies help keep enzymes active, ensuring the cell can keep harvesting energy and building new molecules. Archaeal cells can be extremely small, which can be advantageous in nutrient-poor waters because small cells need fewer resources. A smaller body means a higher surface area to volume ratio, which can help with nutrient uptake when food is dilute. Some archaea build surface structures that help them adhere to particles or other cells, anchoring them in favorable microhabitats. Attachment can keep them near steady nutrient sources or within communities where metabolite exchange supports survival. Certain archaeal communities contribute to methane cycling in permafrost and sediments, linking microscopic metabolism to global greenhouse gas dynamics. When conditions allow, methanogens can generate methane that may accumulate and later be released, connecting tiny cellular processes to large G. Environmental outcomes. Some archaea can incorporate unusual lipid patterns that serve as chemical signatures used to detect them in environmental samples. Because these lipids can be distinctive, scientists can sometimes infer archaeal presence and activity by analyzing lipid molecules in soils, sediments, or water. The microscopic archaeal world includes both specialists for extreme settings and generalists that thrive in ordinary oceans, soils, and living hosts. This range shows that archaea are not only creatures of extremes, they also quietly participate in everyday ecosystems alongside bacteria. Eukaryotic microbes. Microscopy has shown that cells are not just simple sacs of liquid, but carefully organized spaces where many parts are constantly moving and changing. Even in a single tiny cell, you can find structured regions, specialized components, and coordinated activity that keeps the cell alive. Watching cells under a microscope reveals that order and motion are built into life at the smallest scales. Light microscopes are limited by diffraction, which means there is a natural blur created by light waves. Because of that blur, classic light microscopy struggles to separate details much smaller than a few hundred nanometers, even if the lens is excellent. This limit shaped cell biology for a long time because many important structures are smaller than that threshold. Fluorescence microscopy lets scientists highlight specific molecules by attaching fluorescent tags that glow when illuminated with the right light. Instead of seeing everything at once, you can make one protein or one structure stand out clearly against a darker background. This makes it possible to map where certain molecules are inside cells and how they move over time. Confocal microscopy improves clarity in thicker samples by rejecting out-of-focus light, almost like looking at a thin optical slice. By scanning layer by layer, it can build a sharper three-dimensional picture than ordinary wide-field fluorescence. This is especially useful when you want to see structures inside clusters of cells or through thicker biological material. Electron microscopy uses beams of electrons instead of visible light, and electrons have much shorter wavelengths. Shorter wavelengths allow much finer resolution, so electron microscopes can reveal extremely small structures that light microscopes cannot separate. This is how researchers can see detailed cell ultrastructure, like membranes, ribosomes, and finely organized internal compartments. Cryo-electron microscopy and cryo-electron tomography image biological material in a frozen, hydrated state, helping preserve structures close to their natural arrangement. Freezing prevents water from forming damaging ice crystals when done properly, so cells and molecules can remain in place. This approach reduces distortions that can happen with harsh chemical treatments and drying, High-resolution cryo-EM imaging of whole cells often works best near the cell periphery because that region tends to be thin enough for electrons to pass through. Electrons cannot easily penetrate thick material, so thickness becomes a key practical limit. Imaging near the edges gives clearer detail and allows researchers to study membrane regions and nearby internal features. Some thick samples can be thinned for cryo-EM using focused ion beam milling, which carefully removes material to create a thin window. This makes it possible to look deeper inside a cell than the natural thin edge would allow. With this technique, internal regions can be revealed without completely destroying the native architecture. Super-resolution fluorescence microscopy refers to a group of methods that get around the diffraction limit by controlling how fluorescent molecules behave. Instead of accepting the blur as final, these techniques use physics, timing, and computation to locate or confine light more precisely. The result is a view of cell structures at scales that once seemed unreachable with light. STED microscopy achieves super-resolution by using a second light beam to suppress fluorescence around a central point, shrinking the region that actually debores. Emits light. It is like tightening a spotlight, so the glowing area becomes much smaller than normal. This can reveal fine arrangements of proteins and membranes that would otherwise merge into a single fuzzy spot. PALM and related localization methods reach super-resolution by activating only a small number of fluorophores at a time and precisely locating gall, each one. By repeating this cycle many times, a detailed map is built from many individually localized points. This turns the problem of blur into a problem of accurate positioning, handled over many frames. Super-resolution methods can reveal nanoscale organization in cells, such as how proteins cluster or arrange along membranes. Structures that looked like smooth lines or uniform patches can turn out to be composed of many distinct small elements. This helps researchers understand not just where something is, but how it is organized at the scale that often matters for function. Correlative microscopy combines different imaging modes such as fluorescence microscopy for molecular identity and electron microscopy for detailed structure. Fluorescence can tell you which protein you are looking at, while electron microscopy can show the exact shapes and membranes around it. Together, they connect what it is with what it looks like in the cell. Cryogenic correlative approaches can pair super-resolution fluorescence with cryo-electron tomography, linking specific labeled molecules to detailed 3D ultrastructure. This is powerful because it can pinpoint where a particular protein sits within the real physical landscape of a cell. It helps bridge the gap between molecular biology and cellular architecture. Advances in detectors, optics, and computation have improved signal extraction, meaning weaker signals can be captured and interpreted more effectively. Better sensors can detect fewer photons, and better algorithms can separate real information from noise. This allows imaging that is gentler on samples and still informative. Microscopy always involves trade-offs between resolution, speed, and sample health, especially in live cells. Bright light can damage cells or bleach fluorescent markers, while slow imaging can miss fast events. Choosing an approach often means deciding what matters most: fine detail, gentle conditions, or real-time dynamics. Time-lapse microscopy allows scientists to watch living processes unfold, like cell division, organelle transport, or changes in microbial movement. Instead of a single frozen snapshot, you get a sequence that shows cause and effect over minutes or hours. This is one way to see how cellular behavior changes naturally over time. Single-cell microscopy can reveal that genetically identical cells do not always behave identically. Small differences in local conditions or random fluctuations in gene expression can lead to different outcomes for neighboring cells. This helps explain why populations can be diverse even without genetic differences. In bacteria, fluorescent tagging has revealed that proteins involved in division assemble into organized structures at the middle of the cell. Instead of being randomly scattered, key division proteins gather where the cell will split. Seeing this directly helps explain how bacteria divide so reliably despite their small size. Imaging has shown that many bacteria position DNA and cellular machinery with surprising spatial precision. Even without a nucleus, bacterial cells can place key components in specific regions. This organization helps ensure that DNA is copied, separated, and inherited correctly when the cell divides. In eukaryotic microbes, microscopy can track vesicles moving along cytoskeletal tracks, showing how internal traffic is directed. Vesicles are small membrane packages used to move materials, and the cytoskeleton acts like an internal roadway system. Watching these movements shows that even tiny cells have highly coordinated logistics. Live imaging can reveal how yeast buds grow, how organelles move into the bud, and how cell polarity is maintained. Budding is not just a bulge forming anywhere it happens in a controlled location with targeted delivery of materials. This makes yeast a useful model for understanding how cells organize growth in a specific direction. Fluorescent reporters can indicate cellular states such as pH, calcium levels, membrane voltage, or oxidative stress. Instead of only seeing shapes, researchers can see chemical conditions changing inside living cells. These indicators make invisible internal shifts visible as changing brightness or color patterns. Microfluidic devices can hold microbes in carefully controlled environments, allowing precise observation while nutrients or drugs are changed in a measured way. These devices can deliver tiny flows and stable conditions that are hard to achieve in ordinary culture plates. This helps scientists test how cells respond to specific shifts without confusing variables. Microbial colony patterns can be imaged to study how growth, diffusion, and chemotaxis create large-scale shapes from small-scale rules. A colony's branching or ring-like patterns can reflect how nutrients spread and how cells move. By imaging these patterns, researchers can infer the hidden dynamics that produced them. Imaging of biofilms can reveal their 3D architecture, including channels and voids that affect how nutrients and water move through the community. Biofilms are not solid blocks, they can have internal pathways. Seeing these structures helps explain how deep layers can remain alive even when the surface is dense. Microscopy has shown that biofilms can contain microcolonies with distinct gene expression compared to neighboring regions. Within the same biofilm, different zones can behave differently, almost like specialized districts. Imaging and molecular labeling together can show where these functional differences appear. Advanced imaging can track how protists capture prey, including how feeding structures form and how engulfment begins. Some protists use grooves, pockets, or flexible membranes to take in other cells. Watching this in motion reveals the step-by-step mechanics of microscopic predation. High-speed imaging can resolve rapid ciliary beating, showing how tiny hair-like structures move in coordinated waves. Cilia can beat so fast that ordinary video blurs the motion, so high-speed capture reveals the true pattern. This coordination is what makes ciliates such efficient swimmers and feeders. Imaging of diatoms highlights how silica deposition is controlled to build intricate shells with species-specific designs. The shell is not random glass. It is patterned and structured. Microscopy helps reveal how biological processes shape mineral material into complex architecture. Polarized light microscopy can reveal ordered structures inside cells, such as crystalline inclusions or aligned fibers. Some cellular materials interact with polarized light in special ways, making them stand out. This method provides information about internal order, not just overall shape. Phasic contrast and differential interference contrast microscopy enhance visibility of transparent cells without staining. Many cells are nearly invisible in plain bright-field light because they do not absorb much light. These techniques convert small differences in thickness or refractive index into visible contrast. Staining techniques can differentiate cell types, highlight DNA, or reveal cell walls, giving quick insight into structure. Stains can bind specific molecules, making certain features stand out clearly. This can be useful for identifying microbes, seeing nuclei, or checking whether a cell wall is intact. In electron micrographs, membranes appear as distinct boundaries that help researchers map organelles and internal compartments. Because electron density differs across layers, membranes can show up clearly. This makes electron microscopy valuable for understanding the physical layout of cells. Cryo methods reduce artifacts that can come from chemical fixation, preserving more native molecular arrangements. Chemical treatments can shrink, swell, or distort structures, while freezing can lock them in place more quickly. This helps ensure that what is seen is closer to how the cell truly looks. Image analysis and segmentation can quantify cell shapes, track cell lineages, and measure protein localization across thousands of cells. Instead of relying only on visual impressions, researchers can turn images into numbers. This makes it possible to compare conditions systematically and detect subtle differences. Automated microscopy platforms can scan many conditions, enabling large-scale experiments across different microbes, drugs, or environments. Automation allows consistent imaging over time and across many samples. This speeds up discovery and helps identify patterns that would be missed by small manual studies. Fluorescence recovery methods can measure how quickly molecules move or exchange, revealing membrane fluidity and binding dynamics. If a region is bleached and then brightens again, the rate of recovery reflects molecular motion. This helps scientists learn how fast components diffuse or how strongly they are held in place. Single molecule tracking can follow individual proteins in membranes, revealing diffusion, confinement, and clustering. Instead of averaging across millions of molecules, it shows what one molecule does at a time. This can uncover behaviors like temporary trapping in domains or joining short-lived clusters. Super-resolution imaging can reveal whether membrane proteins form nanoscale clusters or are evenly distributed. A uniform blur might hide many tiny islands of proteins. Seeing clustering can change how we understand signaling, transport, and cell-to-cell interactions. Correlative approaches can validate whether a fluorescent signal matches a particular ultrastructural feature seen by electron microscopy. This helps avoid misinterpretation, because fluorescence alone might not show the exact shape of what is labeled. The combined view can confirm what structure the label belongs to. In microbial ecology, fluorescent in situ hybridization, or FISH, can label specific taxa inside mixed communities. FISH uses DNA or RNA probes that bind to matching sequences, lighting up targeted organisms. This allows scientists to see where different species sit relative to each other in real space. Imaging has shown that microbial communities often organize by metabolism, with oxygen users near oxygen sources and anaerobes deeper inside. Position becomes a form of strategy, because local chemistry can change sharply over tiny distances. Microscopy helps reveal these invisible gradients through visible spatial patterns. In host tissues, microscopy can visualize microbes relative to host cells, mucus layers, and immune components. This reveals whether microbes are attached, free-floating, clustered, or kept at a distance. Spatial information can be crucial for understanding health and disease. High-content imaging can detect subtle phenotypes caused by mutations or drugs, such as changes in organelle shape or cell cycle timing. Some effects are too small to notice casually, but become obvious when measured across many cells. This makes imaging a powerful tool for screening and discovery. Microscopic observation remains essential for discovering microbial behaviors that are not obvious from genetic sequences alone. DNA can suggest capabilities, but it cannot always predict real behavior in a living environment. Seeing cells move, interact, and respond can reveal surprises that sequences do not show. Many key ideas in cell biology, including organelles, mitosis, and microbial motility, were first recognized through careful microscopy. Before molecular tools existed, seeing patterns and structures was the main gateway to understanding cells. This historical foundation still matters because direct observation often sparks new questions. Modern microscopy increasingly blends physics, chemistry, biology, and computation to expand what can be seen and measured. New methods often rely on precise light control, clever labeling chemistry, and powerful image processing. This integration has made microscopy one of the most innovative areas in studying the microscopic world. Improvements in labeling chemistry allow specific proteins to be tagged with minimal disruption, improving confidence that observed localization is real. If a tag changes how a protein behaves, the image can mislead. Better labeling methods reduce these risks and make observations more faithful. Taken together, modern imaging methods help connect molecular identity, cellular structure, and dynamic behavior across the microscopic world. They let researchers see where molecules are, what cellular landscapes look like, and how things change in time. This creates a more complete picture of life at scales far below ordinary vision. Eukaryotic microbial life includes many protists, fungi, and microalgae, and what unites them is that their cells contain a nucleus. The nucleus is like a protected library for DNA, keeping genetic material inside a separate compartment. This design allows eukaryotic microbes to manage gene expression with extra layers of control and to build complex internal organization. Protists are a very broad category, describing many eukaryotes that are not animals, plants, or fungi in the traditional sense. It's less a single type of organism and more a convenient label for many different lineages. Because of that, protists can look and behave in wildly different ways, even though many are microscopic. Many protists are single-celled, but some form colonies or multicellular structures, showing that protists don't fit one simple lifestyle. A single species might live as separate cells most of the time, yet gather into larger groups under certain conditions. This variety highlights how eukaryotic life can shift between simplicity and complexity, depending on environment and evolution. Many protists move using cilia, flagella, or amoeboid motion, depending on their lineage and habitat. Cilia act like coordinated tiny oars. Flagella can whip or rotate to propel the cell, and amoeboid motion involves flowing and reshaping the cell body. These different movement styles help protists search for food, avoid threats, and find comfortable microenvironments. Ciliates can coordinate thousands of cilia to swim, feed, or crawl along surfaces with impressive control. The coordinated beating can create directed water currents that bring food particles toward the cell. This turns the cell's surface into both a propulsion system and a feeding system, all powered by microscopic molecular motors. Some protists feed by engulfing prey cells, wrapping them in membrane and bringing them inside for digestion. This process is a bit like swallowing, but on a cellular scale, where the prey ends up in an internal compartment. Enzymes then break the prey down into usable nutrients, fueling growth and movement. Many protists are important predators of bacteria, and this grazing influences bacterial population sizes in many ecosystems. When protists eat bacteria, they also recycle nutrients, turning bacterial biomass into forms that can re-enter the food web. This microscopic predation helps regulate community balance and nutrient flow in water and soils. Some protists are photosynthetic, using chloroplasts to convert light and carbon dioxide into sugars. This means they can act as tiny primary producers, feeding themselves with sunlight rather than hunting other cells. In many waters, these photosynthetic protists contribute to oxygen production and provide energy to larger organisms up the food chain. Diatoms are microalgae, known for building intricate glass-like cell walls made mostly from silica. Silica is the same basic material found in sand, yet diatoms shape it into delicate shells with fine patterns. These shells can protect the cell and also influence how it floats, sinks, and interacts with water currents. Diatom shells often show species-specific patterns, meaning different diatoms have their own recognizable designs. These patterns can be used in ecology to identify which species are present in a sample. Because diatom shells can persist after the cell dies, they can also be used to study environmental history and sediments. Many diatoms reproduce asexually, but some also have sexual cycles that reset cell size and increase genetic diversity. In some diatoms, repeated asexual division can lead to gradual size changes across generations. Sexual reproduction can restore typical size and shuffle genes, helping populations adapt to new conditions. Some diatoms have a slit-like structure called ARAF that helps them glide across surfaces. Instead of swimming freely, these diatoms can move along substrates by secreting substances and using the RAF as part of the movement mechanism. This lets them explore surfaces for nutrients and light in shallow waters or on sediments. Dinoflagellates often have two flagella arranged to produce a spinning motion as they swim. This spinning can help them steer and maintain position in changing water conditions. Their movement style can allow them to migrate up and down through water layers, sometimes following light and nutrient gradients. Some dinoflagellates are photosynthetic, while others are predatory or use both strategies depending on conditions. A mix of feeding modes can be useful in environments where light or prey availability changes. By shifting behavior, dinoflagellates can survive in a wider range of situations than a specialist that relies on only one energy source. Some protists build mineralized coverings, adding even more diversity to microscopic architectures. These coverings can be made from minerals like calcium carbonate or silica, and can provide protection or structural support. They also contribute to sediment formation when the organisms die and their coverings accumulate. Certain protists form spores or cysts that help them survive unfavorable times such as drought, cold, or lack of food. A cyst is like a protective resting package that can endure stress until conditions improve. When the environment becomes suitable again, the protist can exit the cyst and return to active life. Slime molds include stages where many nuclei share one large cell-like mass, showing unusual solutions to growth and movement. Instead of staying as separate individual cells, they can merge into a single, spreading form. This allows them to move and feed in coordinated ways that look almost like a single organism, even though many nuclei are involved. Some slime molds form fruiting bodies that release spores, demonstrating complex life cycles without being animals or plants. The fruiting body helps lift spores into air currents for dispersal. This combination of mobile feeding stages and spore-producing stages allows slime molds to exploit resources and spread to new places. Many parasitic protists have specialized structures that help them invade host cells and obtain nutrients. These structures can attach, penetrate, or manipulate host cell membranes. Such specialization is one reason parasitic protists can be very effective at surviving inside hosts, even though host defenses try to remove them. Some protists have reduced organelles because they rely on host environments for certain metabolic needs. If a parasite can borrow resources from the host, it may not need to maintain the full machinery to produce those resources itself. Over time, this can lead to simplified internal structures that reflect a dependent lifestyle. Microfungi include yeasts and molds, and they can be unicellular or filamentous depending on the species and conditions. Yeasts often live as single cells, while molds often grow as branching filaments. Both lifestyles allow fungi to exploit different kinds of resources in different environments. Yeasts commonly reproduce by budding, where a small outgrowth forms, enlarges, and eventually separates as a daughter cell. Budding is a controlled process, with the cell directing building materials to the budding site. This method lets a yeast population grow steadily, one bud at a time. During yeast budding, the nucleus divides, and one daughter nucleus is guided into the growing bud. This ensures that the daughter cell receives a full set of genetic instructions. The timing and positioning are tightly controlled so that division and bud formation stay coordinated. Yeast cells contain organelles such as vacuoles that help recycle components and manage internal chemistry. A vacuole acts like a storage and recycling center, holding enzymes and maintaining chemical conditions. This helps the cell break down old materials and reuse them, especially during stress or limited nutrients. Vacuoles contain enzymes that can break down proteins, nucleic acids, and other molecules into reusable building blocks. This internal breakdown is crucial for cellular housekeeping and survival. When nutrients are scarce, recycling inside vacuoles can supply the cell with needed components without requiring new external food. Yeast cytoskeleton structures help position organelles and coordinate division, and microtubules play a major role during mitosis. The cytoskeleton is like an internal framework and transport system, helping move parts to the right place at the right time. Without it, budding and nuclear division would be less reliable and less orderly. In yeast reproduction and mating, organelle inheritance and mixing can follow carefully timed patterns. The cell does not simply scatter organelles randomly. It often ensures essential structures are delivered to new cells. This helps maintain healthy daughter cells and supports successful reproduction. In early yeast zygotes, some organelles can intermingle without immediately fusing, showing that organelle behavior can be controlled rather than automatic. This separation can matter because organelle fusion affects how genetic material in organelles is inherited. Controlled mixing allows cells to manage inheritance and compatibility more carefully. Many molds grow as networks of hyphae microscopic filaments that extend and branch across a substrate. This growth style allows fungi to explore a wide area and reach scattered nutrient sources. A hyphal network can act like a living web, extending outward where resources are richest. Hyphal growth lets fungi penetrate substrates and absorb nutrients over large surface areas. Instead of swallowing food, fungi often release enzymes outside the body, break material down, and absorb the simpler products. Hyphae provide the physical reach and surface area needed to make this approach efficient. Fungal cells have cell walls rich in chitin and other polymers, giving strength while still allowing growth. Chitin is a tough structural molecule also found in some animal exoskeletons. In fungi, the wall provides protection and shape, while growth occurs at specialized regions where new wall material is added. Some fungi form spores specialized for dispersal through air, water, or animals. Spores can be light and durable, making them effective traveling forms. By producing spores, fungi can spread far beyond the original colony and colonize new environments when conditions allow. Microalgae include many lineages beyond diatoms, and they often form major components of plankton communities. These tiny photosynthetic organisms drift in water and capture sunlight. turning it into chemical energy. They are important not only for themselves, but for the many organisms that feed on them. In oceans, microscopic photosynthetic eukaryotes contribute substantially to global oxygen production and carbon fixation. By using sunlight to convert carbon dioxide into organic matter, they help regulate atmospheric chemistry. Over time, their collective activity has a major influence on how carbon cycles through the planet. Many eukaryotic microbes have mitochondria, organelles that generate ATP using controlled electron transport. ATP is a cell's main energy currency, and mitochondria are specialized for producing it efficiently. By managing electron flow and gradients, mitochondria help power everything from movement to growth. Mitochondria and chloroplasts typically have double membranes, which fits with the idea that they originated through an engulfment event. The outer membrane can be seen as the boundary created during engulfment, while the inner membrane reflects the original membrane of the captured bacterium. This structure supports the broader understanding of how these organelles became integrated into eukaryotic cells. Mitochondria usually carry their own DNA, often in a circular form, separate from nuclear DNA. This means mitochondria retain a small genetic toolkit of their own, even though many mitochondrial proteins are encoded in the nucleus. The existence of mitochondrial DNA is one of the clues pointing to their bacterial ancestry. Many chloroplasts also carry their own DNA and replicate by division resembling binary fission. This replication style is similar to bacterial reproduction, where one unit divides into two. It shows that chloroplasts, like mitochondria, have retained bacterial-like traits even while functioning as organelles inside eukaryotic cells. Mitochondria and chloroplasts have ribosomes that resemble bacterial ribosomes more than typical eukaryotic cytosolic ribosomes. Ribosomes are protein-building machines, and these organelle ribosomes reflect their evolutionary origin. This difference also matters in medicine, because some antibiotics affect bacteria-like ribosomes more than eukaryotic ones. Some protists possess unusual mitochondrial variants adapted to low-oxygen environments. When oxygen is scarce, standard mitochondrial respiration may not work well, so these variants can use alternative chemistry. This adaptation lets some eukaryotic microbes survive in sediments or other oxygen-poor habitats. Many eukaryotic microbes regulate gene expression through chromatin structure, using DNA packaging proteins to control access to genes. DNA is wrapped and organized, and how tightly it is packed can determine whether a gene can be read. This adds a level of control beyond simply having a gene present, allowing fine-tuning of which proteins are made. The nucleus separates transcription from translation, meaning RNA is made in the nucleus while proteins are built outside it. This separation enables additional regulation, such as RNA processing, editing, and controlled export. It also allows the cell to coordinate when and where messages are translated into proteins. Many eukaryotic microbes use vesicle trafficking to move proteins and lipids between organelles. Vesicles are tiny bubbles of membrane that carry cargo, ensuring materials reach the correct destination. This internal shipping system is essential for maintaining specialized organelles and for secreting substances outside the cell. The endoplasmic reticulum and Golgi apparatus work together to produce and modify proteins destined for secretion or membranes. The ER helps build and fold proteins, while the Golgi further processes and sorts them. This assembly line ensures proteins end up in the right place and with the right chemical modifications. Many protists and fungi secrete enzymes outside the cell to digest food externally before absorption. This strategy allows them to access nutrients in large particles or complex materials without engulfing them. After enzymes break the material down, the cell absorbs smaller molecules through its membrane. Some eukaryotic microbes produce protective pigments that help manage light stress or oxidative damage. Too much light can create harmful reactive molecules, so pigments can act like shields and filters. These pigments can also influence the color of microbial blooms and communities in natural waters. Many microscopic eukaryotes can shift between feeding modes, photosynthesis, predation, or absorption, depending on what the environment offers. This flexibility is helpful in changing conditions, such as cloudy days, nutrient pulses, or fluctuating prey availability. By switching strategies, they can survive where specialists might fail. Protists can range from about 1 micrometer to several millimeters, and some related groups include forms that are much larger. This size range means microscopic can still include organisms visible as tiny specks under the right conditions. Even so, many protists remain small enough that their worlds are shaped by surface tension, diffusion, and microscopic currents. The microscopic eukaryotic world contains immense diversity in cell structure, movement, feeding, reproduction, and ecological roles. Some act as primary producers, some as predators, some as decomposers, and some as parasites. Together, they show how much complexity and variety can exist even within a single cell. Many microbes live in mixed communities where bacteria, archaea, and eukaryotic microbes all interact, often within the same small patch of water, soil, or host tissue. In these settings, no single organism is truly living alone because neighbors change the chemistry around them. The result is a microscopic neighborhood where each species can affect what others can eat, how they grow, and which stresses they can survive. Microbial communities often exchange metabolites, meaning one organism's byproducts become another organism's nutrients. What looks like waste to one microbe can be valuable fuel to another, so materials keep circulating instead of being discarded. This sharing can make communities more efficient overall, allowing them to extract more energy from the same resources. In anaerobic environments, hydrogen buildup can block some energy pathways, because too much hydrogen changes the balance of chemical reactions. Partner organisms that consume hydrogen can remove this bottleneck, making it easier for other microbes to keep producing energy. This kind of cooperation is like keeping a system from clogging, so the whole community can continue functioning. In the human gut, microbial fermentation of dietary fiber often produces short-chain fatty acids, such as acetate, propionate, and butyrate. These molecules are created when microbes break down complex plant material that human enzymes can't fully digest. In this way, gut microbes turn fiber into smaller compounds that can influence both the gut environment and the body as a whole. Short-chain fatty acids can be absorbed by the host and can act as signaling molecules, meaning they do more than just supply energy. They can influence how gut cells behave and how certain immune and metabolic pathways respond. This shows how microbial chemistry can reach beyond the microbes themselves and interact with the host's broader physiology. The human gut microbiome includes bacteria, archaea, and fungi, each contributing different abilities. Bacteria often dominate in number, but archaea and fungi can still play meaningful roles in gas chemistry, fermentation balance, and competition. Thinking of the microbiome as a multi-domain ecosystem helps explain why gut function is influenced by more than one type of microorganism. Some gut-associated archaea consume hydrogen and produce methane, which changes local gas balance and affects fermentation dynamics. By removing hydrogen, they can indirectly support other fermenting microbes that would slow down if hydrogen accumulated. Their methane output is a visible reminder that microbial metabolism can create gases as natural products. Microbial ecosystems can behave like layered chemical factories, where different zones carry out different steps of a larger process. Near oxygen sources, oxygen-using microbes often dominate, while deeper zones may be oxygen-free and support anaerobic metabolism. This layering can happen over millimeters or even less, creating sharply different microworlds stacked together. Many microbes compete by producing antibiotics or other inhibitory compounds that reduce the growth of rivals. In the wild, these chemicals can help a microbe protect its food supply or territory. Competition at the microscopic scale can be intense, and chemical warfare is one tool microbes use to influence who gets to survive nearby. Some microbes resist antibiotics through enzymes that destroy or modify the drug, pumps that push drugs back out of the cell, or altered targets that drugs can no longer bind effectively. These strategies work like different kinds of shields. One breaks the weapon, one ejects it, and one makes it miss. This variety is one reason antibiotic resistance can be difficult to overcome. Microbial resistance genes can spread through horizontal gene transfer, meaning resistance can move between cells rather than waiting for slow mutation. A resistance gene carried on the plasmid can pass from one microbe to another, potentially across species boundaries. This helps explain why resistance can rise rapidly within communities exposed to antibiotics. Many microbes use iron scavenging molecules to capture scarce iron because iron is essential but often difficult to access. These molecules can bind iron tightly and bring it back to the cell, almost like specialized collectors. Because iron is so valuable, competition for it can shape which microbes thrive in a given habitat. Some microbes store nutrients internally as granules, creating reserves for times when the environment becomes less generous. This storage can include carbon-rich compounds or other key resources needed for survival. Having internal archaea reserve lets microbes keep functioning through short periods of scarcity. Many microbes coordinate stress responses using molecular chaperones that help proteins fold correctly and avoid damage. When conditions like heat or toxins threaten protein shape, chaperones can stabilize or refold proteins. This is crucial because proteins do most cellular work, and damaged proteins can disrupt many processes at once. Heat shock and oxidative stress systems can rapidly reshape cellular priorities, shifting focus toward protection and repair. Instead of investing energy in growth, the cell may temporarily prioritize fixing damage, removing harmful molecules, and producing protective proteins. This flexibility helps microbes survive sudden changes like temperature spikes or chemical stress. Many microbes maintain internal pH by moving protons and other ions across membranes, keeping their internal environment stable. If the outside becomes more acidic or more alkaline, cells must compensate to protect enzymes that only work in a narrow pH range. Ion transporters and buffers work together to keep internal chemistry suitable for life. Osmorgulation in microbes often involves accumulating compatible solutes, small molecules that balance water pressure without disrupting Banach. Proteins. This helps cells avoid shrinking in salty environments or bursting in fresher conditions. Compatible solutes act like safe internal ballast, adjusting water movement while protecting delicate cellular machinery. Some microbes withstand desiccation by forming protective sugars and proteins that stabilize membranes and DNA when water is scarce. Without water, cellular structures can collapse or break, so these protected molecules act like stabilizing supports. This ability helps microbes survive drying on rocks, soils, or surfaces exposed to air. UV radiation can damage DNA, and many microbes have efficient repair pathways that fix UV-induced lesions. Sunlit environments can be harsh at the molecular level, even when they look calm to us. DNA repair helps microbes maintain genetic integrity and continue reproducing despite frequent exposure to damaging radiation. In nutrient-poor oceans, Some microbes have streamlined genomes and low resource demands, allowing them to survive on very small amounts of available food. Streamlining can mean fewer genes, simpler regulation, and fewer costly structures. This approach can be very successful when nutrients are dilute and competition is fierce. In richer environments, microbes may carry larger genomes with diverse metabolic options, letting them take advantage of many different resources. If one food source disappears, they can switch to another. This flexibility can be beneficial in habitats like soils or guts, where chemical conditions and available nutrients change frequently. Many microbes switch between fast growth and survival states depending on resources. When nutrients are plentiful, cells may prioritize rapid division. But when food becomes scarce, they may slow down and focus on maintenance and repair. This shift helps microbes persist through cycles of feast and famine. Some microbes produce extracellular enzymes that break down polymers outside the cell, creating shared nutrients that others can also use. This can be efficient because one enzyme can release many nutrient molecules from a large substrate. But it also creates a social challenge. Nearby microbes may benefit without paying the cost of producing enzymes. Cooperation and cheating can both occur in microbial communities because shared resources invite both contributors and freeloaders. Some cells invest energy to produce public goods, while others exploit those goods. The balance between cooperation and cheating can shape how stable a community remains and how it evolves over time. Microbes can form spatial patterns where different species settle into micro-niches defined by oxygen, light, pH, and nutrients. Even tiny distance differences can matter because chemicals diffuse and are consumed quickly. These micro-niches allow many species to coexist by specializing in slightly different conditions. Even within one biofilm, cells can differ in growth rate, gene expression, and stress tolerance, depending on where they sit. Cells near the surface may get more oxygen or nutrients, while deeper cells may grow slowly and become more stress-resistant. This internal variety can help the biofilm survive changing conditions. The extracellular matrix in biofilms helps communities adhere to surfaces and resist being washed away. It also acts like a protective layer that can slow down the penetration of harmful substances. This matrix is one reason biofilms can persist on rocks, pipes, teeth, and many other surfaces. Extracellular polymeric substances can form a large fraction of biofilm organic matter and strongly influence biofilm physical properties. These substances give the biofilm a gel-like structure that can hold water and bind particles. The composition of the matrix affects how strong, sticky, or porous the biofilm becomes. The biofilm matrix can trap nutrients and retain water, creating a more stable microenvironment for cells inside. This can buffer microbes from sudden drying or nutrient fluctuations. In effect, the biofilm becomes a self-made habitat that smooths out environmental extremes. In soils, microbial extracellular polymers can help bind particles, improving aggregation and influencing how water moves through the soil. When soil aggregates are stable, they can hold water and air in a balanced way. Microbial polymers contribute to soil structure, linking microscopic secretions to large-scale soil health. Microbial communities can influence plant health by cycling nutrients near roots and competing with plant pathogens. Healthy root-associated communities can help make nutrients available and can occupy space that pathogens might otherwise take. This means the microscopic world around roots can strongly shape plant growth and resilience. Some fungi form symbioses with plant roots, extending networks into soil that gather nutrients in exchange for plant sugars. These fungal networks can reach into tiny soil spaces that roots alone cannot explore well. In return, plants feed the fungi with carbon compounds, creating a mutually beneficial trade. Some bacteria live inside plant tissues and can contribute to nutrient availability or stress tolerance. Inside the plant, they may help supply nutrients, produce growth-supporting compounds, or assist in responding to environmental stress. This kind of close association shows how microbes can become part of a plant's extended survival system. In aquatic systems, microbial loops recycle dissolved organic matter into forms that can re-enter the food web. When small organic molecules are released into water, microbes can capture them and convert them into microbial biomass. That biomass can then be eaten by microscopic predators, moving energy upward. Many microscopic predators, including protists, help control bacterial populations and recycle nutrients by grazing. When protists eat bacteria, they convert bacterial carbon and nutrients into their own biomass and release some back into the environment. This grazing helps prevent any single bacterial group from dominating unchecked. Some microbial interactions involve direct cell-to-cell contact, including close associations where partners exchange nutrients or signals. Physical contact can make transfer more efficient, ensuring molecules reach the partner quickly. In some cases, contact-dependent interactions can also enable competition or defense strategies. Microbes can communicate indirectly by altering shared chemistry, such as changing pH, oxygen levels, or nutrient availability. Even without direct signals, one species can reshape the environment in ways that help or harm others. This means the conversation between microbes can happen through the environment itself. Some microbes produce gases such as methane, carbon dioxide, hydrogen sulfide, or nitrous oxide, linking cellular metabolism to atmospheric ED. Chemistry. These gases are natural metabolic products, especially in oxygen polar settings. When released in large amounts across ecosystems, they can influence larger-scale environmental processes. Microbial activity in sediments often occurs in layers, with oxygen consumed near the surface and anaerobic metabolisms dominating deeper down. As oxygen disappears with depth, different energy pathways take over. This layering creates a vertical map of metabolism, with different microbes thriving at different depths. Many microbes can use nitrate, sulfate, or carbon dioxide instead of oxygen in energy generation. These alternative electron acceptors allow life to continue where oxygen cannot reach. The choice of acceptor affects which end products form, shaping the chemistry of environments like sediments and wetlands. Microbial metabolism can mobilize or immobilize metals, influencing water quality and mineral behavior. By changing metal oxidation states, microbes can make certain metals dissolve more easily or become locked into solid minerals. This can affect contamination, nutrient availability, and the formation of mineral deposits. Some microbes precipitate minerals that preserve microstructures, leaving microscopic signatures that can persist over long times. When minerals form around cells or biofilms, they can capture shapes and textures. These preserved patterns can become part of the geological record, providing clues about past microbial life. Symbiosis can be so tight that some microbes lose genes they no longer need, relying on partners for missing functions. This gene loss can streamline the organism, but also makes it dependent. Over evolutionary time, such dependence can lead to highly specialized microbes that cannot easily live alone. Some host-associated microbes are passed from parent to offspring, helping stabilize communities across generations. When transmission is reliable, the host and microbe can develop long-term relationships. This can create a stable partnership where both sides benefit from predictability. Microbial community composition can shift rapidly with diet, temperature, moisture, or chemical exposure because microbes respond quickly to changing stimuli. Resources. A new nutrient source can favor certain species, while stress can reduce others. These shifts can happen much faster than changes in larger organisms, reflecting microbes' rapid growth and adaptability. Some microbes specialize in breaking down particular food components, like certain fibers, and their abundance can rise when those foods increase. This is a direct example of ecology in action. Supply changes demand, and populations adjust. Over time, the available diet can shape which microbial functions become common. Microbial ecosystems can show resilience, sometimes returning toward prior states after disturbances, though not always completely. After a disruption, some species may regrow and re-establish a familiar balance, while others may remain changed. This resilience depends on many factors, including which microbes survive and what conditions follow. Many microbial communities are shaped by both which organisms are present and which genes they carry, because gene exchange can add new abilities without crude, changing the species list. A community can gain resistance or new metabolism if key genes spread through horizontal transfer. This makes microbial ecosystems highly dynamic at the functional level. The microscopic world includes not only single-species behaviors, but also multi-species networks that act like integrated systems. Different organisms can form chains of metabolism, share protection, or shape habitats together. These networks can behave like a single unit in some ways, responding collectively to changes. Across land, oceans, and living bodies, microbial interactions drive nutrient cycles, stabilize ecosystems, and influence chemistry on large scales. Tiny cells collectively transform massive amounts of matter and energy each day. Their combined activity connects the microscopic and the planetary, showing that small life can have enormous impact. Microbial life cycles can include multiple forms, such as active growth stages and dormant stages built for survival and dispersal. In active stages, cells focus on feeding, dividing, and expanding their population. But when conditions become harsh, too dry, too cold, too nutrient poor, they may shift into a protected state. This ability to pause and later resume is one reason microbes can persist through seasons, disasters, and long stretches of difficulty. Many microbes sense chemical gradients and adjust their swimming or crawling to move toward favorable conditions, a behavior often called chemotaxis. They do this by detecting whether chemical signals are increasing or decreasing as they move, then altering direction to improve their chances of finding nutrients. Even without a brain, this simple guidance system helps microbes navigate tiny landscapes where food may be patchy and short-lived. Some bacteria can alternate between planktonic, free-swimming lifestyles and surface-associated biofilm lifestyles, depending on environmental cues. When nutrients are scattered in water, swimming can help them search widely. But when a good surface is found rich in food or stable conditions settling down and forming a biofilm can be safer. This switch allows the same organism to behave like an explorer at one moment and a community builder at another. The same microbial species can express different genes in different habitats, making its behavior highly context-dependent. In one place, a microbe may turn on genes for movement and nutrient scavenging, while in another it may invest in protective layers or stress tolerance. This flexible gene control helps microbes match their biology to local conditions instead of behaving the same way everywhere. Microbes often produce enzymes that help them exploit specific niches, such as breaking down unusual sugars or resistant polymers that other organisms cannot easily use. Enzymes act like specialized chemical tools, cutting complex molecules into smaller pieces that can be absorbed. By carrying unique enzymes, A microbe can thrive on resources that few competitors can access. Some microbes rely on energy sources that are scarce or spread out, so they require very efficient uptake systems to survive. When a nutrient is extremely dilute, a cell needs transport proteins that capture it quickly and hold onto it. This is like having very sensitive molecular fishing lines that snag rare molecules before they drift away. Microbial cells must constantly manage oxidative stress because reactive oxygen species can damage DNA, proteins, and lipids. Reactive oxygen can be produced by normal metabolism, by sunlight, or by certain chemicals in the environment. If left unchecked, it can disrupt cellular machinery, so managing oxidative stress is a daily maintenance task for many microbes. Many microbes deploy antioxidant enzymes and repair systems to keep oxidative damage from becoming overwhelming. These systems can neutralize reactive molecules, repair damaged DNA, and remove proteins that have become dysfunctional. The goal is not to avoid all damage because some is unavoidable, but to keep damage low enough that the cell remains functional. Some microbes can tolerate high radiation by repairing DNA breaks rapidly and protecting proteins from oxidation. Radiation can break DNA strands, which is a severe form of damage, but certain microbes have repair strategies that reassemble broken DNA efficiently. They may also use protective molecules that reduce protein damage, helping the cell keep working while repairs are underway. In cold environments, some microbes adjust membrane composition and produce antifreeze like proteins to keep cellular processes running. Cold can stiffen membranes and slow chemical reactions, so cells often increase membrane fluidity and protect against ESA-related stress. These adjustments help nutrients still cross membranes and enzymes still function, even when temperatures are low. In hot environments, microbes often rely on heat-stable proteins and chaperones that prevent unfolding and clumping. Heat can make proteins lose their shape, and misfolded proteins can stick together in harmful aggregates. Chaperones help keep proteins properly folded, allowing the cell's machinery to continue operating under thermal stress. Microbes living at high pressure may adjust membranes and enzyme structures to function under compression. High pressure can affect how molecules fit together and how membranes behave, so pressure-adapted microbes often have subtle biochemical features that keep systems flexible and active. This helps them survive in deep-sea environments where pressure is far beyond what surface life experiences. Some microbes live in tiny water films on soil particles, where moisture and nutrients can change rapidly. In these thin films, conditions can shift from wet to dry within hours, and nutrients can appear in brief pulses. Microbes adapted to this lifestyle must respond quickly, capturing food fast and enduring drying periods when the film disappears. Microbial activity can surge after rain because water reconnects habitats and carries dissolved nutrients. Rain can dissolve and transport organic matter, making it suddenly available to microbes that were previously limited by dryness or isolation. This can lead to bursts of growth and metabolism, where microbes quickly process newly accessible compounds. In oceans, microscopic plankton communities shift with seasons, light levels, and nutrient mixing, reshaping food webs. When sunlight increases, photosynthetic microbes may bloom, and when nutrients rise due to mixing, different groups may take advantage. These changes can cascade upward, affecting the organisms that feed on plankton and the larger ecosystem structure. Some microalgae can form resting stages that sink or persist until conditions become favorable again. When light or nutrients drop, entering a resting stage can help them survive rather than die off completely. Later, when conditions improve, these resting forms can return to active growth, helping populations rebound. Many microbes emit or respond to volatile compounds, enabling chemical interactions that can extend beyond direct contact. Volatile compounds can travel through air or water and act as signals, warnings, or competitive tools. This means microbes can influence one another even when they are not touching, through chemical messages that drift through the environment. Microbes can influence each other's evolution by creating selective pressures, such as producing toxins that favor resistant neighbors. When one microbe produces a harmful compound, only those with resistance mechanisms will survive nearby. Over time, this can shape the genetic makeup of populations and drive the spread of protective traits. Some bacteria produce bacteriocins, which are narrow-spectrum toxins that often target closely related strains. This allows a bacterium to reduce competition from near relatives that share the same niche and resources. Because bacteriocins can be specific, they help shape fine-scale community structure, deciding which strains dominate a microhabitat. Microbial cells often prioritize energy efficiency, using regulatory circuits that avoid making expensive proteins when they are not needed. Protein production costs energy and raw materials, so turning on genes only when useful helps conserve resources. This efficiency can be critical in environments where nutrients are scarce and survival depends on careful budgeting. Some microbes show a kind of division of labor, where subsets of cells specialize in particular roles, especially in structured communities. One group might focus on producing protective substances, while another focuses on growth or resource capture. Specialization can improve overall performance, because not every cell must do every job at once. In some bacterial communities, a fraction of cells may produce large amounts of extracellular matrix, while others focus on growth. Matrix producers create the protective and supportive structure that benefits the whole community, while fast-growing cells expand population size. This balance can make a community both resilient and able to spread. Some microbial ecosystems show succession, where early colonizers change conditions and enable other species to arrive later. Early microbes might consume oxygen, alter pH, or produce metabolites that become food for new species. Over time, the community composition can shift in an orderly way, reflecting how the environment is being remodeled, step by step. In host-associated settings, microbes often must tolerate immune defenses while also competing with each other. The host environment can be stable and nutrient-rich, but it also includes barriers, antimicrobial molecules, and immune cells. Microbes that persist there must balance stealth, resilience, and competition all at once. Many microbes produce surface molecules that help them adhere to specific tissues, particles, or other cells. Attachment can keep them in a favorable location where nutrients flow past, or help them avoid being swept away. These surface features can also influence which partners they can interact with and which hosts they can colonize. Microbes can form mutualisms where partners exchange nutrients, protection, or services like detoxification. In such partnerships, each participant gains something it would struggle to obtain alone. Mutualisms can stabilize communities and allow microbes to occupy niches that would otherwise be too harsh or too resource-limited. Some protists host internal symbionts, including photosynthetic partners that supply sugars in exchange for shelter and nutrients. The protist provides a stable home and access to certain resources, while the symbiont converts sunlight into usable energy-rich molecules. This is a cellular partnership, where two organisms effectively share one living space. Some fungi host intracellular bacteria, showing that cells within cells relationships can occur across domains. The bacteria may influence fungal metabolism, survival, or interactions with the environment. These nested relationships illustrate how cooperation and dependence can evolve even between very different kinds of life. Microbial communities can be shaped by physical forces like flow and shear, which affect which cells can stay attached. In fast-flowing water, only strongly attached organisms or robust biofilms may persist. Physical forces can therefore act like an environmental filter, selecting for certain surface traits and community structures. In streams and pipelines, biofilms can grow as complex layered structures that respond to changing flow and nutrient supply. When flow brings more nutrients, growth may accelerate, while strong shear forces can reshape the biofilm surface. Over time, these structures can become highly organized, reflecting both biology and physics working together. Many microbes can sense surfaces and switch on genes that increase adhesion and community formation. Touching a surface can be a signal that a stable habitat is available. In response, cells may produce sticky molecules, reduce swimming, and begin building the structures needed for a long-term community. Some bacteria produce surfactants that change surface tension, influencing spreading and colony shape. Surfactants can help bacteria move across surfaces more easily and can alter how droplets and films behave. By changing the physical environment, surfactants can help bacteria expand their territory or access new resources. Microbes often use small RNAs and regulatory proteins to fine-tune gene expression quickly under stress. These regulators can act faster than building new proteins from scratch because they can adjust existing messages and control translation. This rapid control helps microbes respond to sudden shifts like heat, toxins, or nutrient loss. Many microbes store information not only in DNA but also in epigenetic marks that affect how genes are expressed. Epigenetic marks can influence which genes are active without changing the DNA sequence itself. This can provide a flexible layer of memory, helping cells respond differently based on recent conditions. Microbial genomes can be dynamic over evolutionary time, with gene gains, losses, and rearrangements changing what a microbe can do. If new genes arrive through gene transfer, new metabolic abilities can appear quickly. If genes are lost, the microbe may become more specialized, focusing on a narrower lifestyle. Some microbes maintain multiple copies of certain genes to boost production of crucial proteins when needed. Having extra copies can increase output, especially if a protein is essential for growth or stress survival. This is one way microbes can amplify key functions without inventing entirely new systems. Many microbial proteins assemble into complexes that work like nanoscale machines, performing tasks such as transport, energy conversion, and movement. These machines are built from multiple parts that fit together and move in coordinated ways. They show that mechanical-like function can exist even at molecular scales inside a single cell. Ribosomes translate genetic information into proteins by reading messenger RNA and linking amino acids in the correct order. This process is the bridge between genetic instructions and physical cellular machinery. Because proteins do so many jobs, ribosome function is central to growth, repair, and every aspect of cellular life. The genetic code is largely shared across all cellular life, meaning the same language of codons and amino acids is used in bacteria, archaea, eukaryotes. This common code allows the same basic rules of translation to apply across life. It is one reason scientists can study fundamental biology in many different organisms and still find shared principles. Despite shared principles, microbes differ greatly in their metabolic toolkits, reflecting the niches they occupy. Some are built to harvest light, some to oxidize minerals, some to ferment sugars, and some to live on very unusual chemicals. This diversity in metabolism is one reason microbes can occupy nearly every environment on Earth. Some microbes can switch metabolic modes using oxygen when it is available, and shifting to anaerobic pathways when oxygen disappears. This metabolic flexibility helps them survive in habitats where oxygen fluctuates, like sediments or biofilms. Switching modes can keep energy flowing even when conditions change suddenly. In many natural settings, microbes operate in energy-limited states, growing extremely slowly but staying active enough to repair damage. Growth may be rare, but maintenance never stops, because molecules degrade over time. By focusing on minimal activity, microbes can persist in environments where energy is just barely sufficient for life. Microbial cells can be harmed by their own metabolic byproducts, so balancing energy production with protection is a constant challenge. For example, producing energy may generate reactive oxygen species or other harmful intermediates. Cells must harvest energy without letting the side effects accumulate faster than they can be managed. Many microbes use feedback loops to stabilize internal conditions, preventing runaway changes in pH, ion balance, or redox state. Feedback loops are like automatic controls. When something shifts too far, the cell triggers responses to push it back toward a safe range. This stability keeps enzymes working and protects vital structures. Microbial diversity is enormous, and many lineages are known only from genetic sequences, suggesting vast, unseen functional variety. DNA surveys reveal branches of life that have not been cultured or observed directly. Each new lineage may represent unique metabolic strategies and ecological roles we are only beginning to understand. Culturing in the lab captures only a subset of microbial life because many organisms depend on specific partners or microconditions that are hard to BS. Reproduce. Some require precise nutrients, unusual gases or chemical gradients, while others rely on community interactions. This is why environmental sampling often reveals far more diversity than lab cultures. Microscopic life includes both long-term stable communities and rapidly changing assemblages that respond to short-lived resources. A stable community might persist in a consistent environment, while a transient community may bloom quickly after a nutrient pulse and then fade. Both patterns are common in nature, reflecting how dynamic microscopic ecosystems can be. Across all domains, cellular life depends on membranes, genetic information, proteins, and energy flow, yet implements these essentials in countless different ways. The basic building blocks are shared, but the engineering solutions vary widely. This is what makes the microscopic world both unified and endlessly diverse. The microscopic world is a continuous interplay of structure, chemistry, ecology, and evolution happening at scales too small to see unaided. Every tiny environment contains chemical gradients, interactions, competition, cooperation, and adaptation. Together, these processes shape ecosystems and life's possibilities, one cell at a time. When you look closely at cells, even the simplest microbe is revealed as a tightly organized system that survives by regulating its internal world, repairing gray, damage, and adapting to change. It doesn't need to be large to be complex, and it doesn't need to be visible to be powerful in its effects. And as you let these quiet details settle in your mind, you can simply allow your attention to soften, knowing that life continues gently at these tiny scales, steady and unhurried, all around you.